Okay, part 2 of herbicides mode of action. The the next mode of action is amino acid biosynthesis inhibitors. Sometimes it's called as ALS. Okay, uh, the acetolactate synthase inhibiting factors. The acetolactate synthase inhibiting herbicides. These herbicides have a broad spectrum of selectivity and are used at low rates as soil applied or post-emergence treatments in a variety of crops. These herbicides inhibit the selectivity of the activity of the ALS enzyme, which is involved in the synthesis of the branch chain amino acids such as leucine, isoleucine, and valine. Remember here, amino acids are essential building blocks in proteins and are required for production of new cells. So, ALS herbicides are readily absorbed by both roots and foliage and translocated in both the xylem and phloem to the site of action at the growing points. The amino acid synthesis inhibition mode of action includes herbicides from the following chemical families. First is sulfonylureas, imidazolinone, and some amino acid derivatives such as triazolopyrimidines, EPSP synthesis inhibitors, and the glutamine synthase inhibitors. The sulfonylureas, imidazolinone, and triazolopyrimidines are also known as ALS or AHAS inhibitors, like I mentioned in the slide before. All of the herbicides within this mode of action act upon specific enzymes to prevent production of amino acids. So amino acids are the building blocks for proteins for plant growth and development of a plant, like I mentioned before. So this is very, very important and crucial. So they cut the production. So this kind of herbicide cut the production of the protein. So these are some examples of amino acid synthesis inhibitors. So sulfonylureas, the core structure of this herbicide group is the combination of the photosynthetic inhibitors, ureas and triazines. But the primary mechanism of action is inhibition of amino acid synthesis, not the photosynthesis. Secondarily, they inhibit photosynthesis, respiration, and protein synthesis. So plant symptoms include chlorosis, necrosis, terminal bud death, and vein discoloration. The site of action for the sulfonylureas catalyzes the first step in the biosynthesis of the three-branch chain aliphatic amino acids, which is valine, leucine, and isoleucine. A secondary effect is cessation of plant growth, such as stunting, due to cessation of cell division and slow plant death. Tolerance is related to plant's ability to detoxify the herbicide. So these are some of the common sulfonylurea herbicides. So clearfield rice is one of rice varieties developed by BASF to be resistant to imidazolinone herbicide, which means that this herbicide clearfield will kill majority of weeds in the rice field, but will not kill the clearfield rice. This clearfield rice. The drawback is at the same time the clearfield will also kill any non clearfield. So like any conventional varieties like MR220, MR219, so MR297, and so on and so forth. So this is a drawback. So let's say there is a drift or runoff from the clearfield field to the neighboring rice field. This will be a problem. This is one of the drawback of this clearfield rice. So injury symptoms caused by ALS inhibiting herbicides are not apparent until several days after treatment, although susceptible plants stop growing almost immediately. Affected plants can exhibit stunting, interveinal chlorosis, chlorotic bending on grass leaves, red leaf venation, purpling, root pruning, and gradually death. The risk of crop injury from the sulfonylurea and sulfonamide herbicide is more likely on high than low pH soils. So the effect, the crop injury is more effective on high pH soils. Okay, so these are the injuries. So I already give you some notes. So in the transcript, so you can read and then pause this video and then try to understand these herbicide injuries. Photosynthetic inhibitors. The two photosynthetic reactions are coupled by the photosynthetic electron transport chain, where photophosphorylation, which is occurs. Photophosphorylation is what? The production of ATP. So photosystem 1 produces reduced nicotine adenine dinucleoside phosphate, the NADP. So herbicides that act in relation to photosystem 1 divert electrons away from photosystem 1 and generate a toxic molecular species. So in photosystem 2, where most herbicides in this group act, begins with removal of electrons from water and production of oxygen, which in the Hill reaction. Okay, most of these large and diverse group of chemical structures that inhibit photosystem 2 block electron transport by binding to adjacent sites on the D1 quinone protein of the photosynthetic system. So the functions in the electron transport chain between the primary electron acceptor from chlorophyll and plus 2 quinone. This is how the photosynthesis inhibitors react or function in the photosynthesis systems. The chemical groups includes amide, the one herbicide, or benzothiazole, nitrile, phenyl carbamate, phenyl pyridazine, pyridizinone, triazinone, uracil, and urea. More herbicides chemical groups act on photosynthesis than on any other physiological processes. A summary of selectivity information on some of these herbicides is in this table. So look at this table and look to the information. These herbicides in general cause gradual chlorosis in plants. So pause this video to look at the description on the symptoms and on the injuries. Next is cell membrane disruptor. Herbicides in this group are often called photodynamic. They act independently of photosynthesis but require light for activity. These herbicides inhibit the enzyme protoporphyrogen oxidase, the PPO or Protox, a step in the porphyrin pathway that produces half of the chlorophyll molecule. Unlike Protox inhibitors cause accumulation of large amounts of the phytotoxic molecule protoporphyrin or proto. Proto accumulation quickly damages lipids and proteins, resulting in chlorophyll and carotenoid pigment loss and leaky membranes. For instance, the plasma, lemma, and the tonoplast that lead to cell desiccation and disintegration. Herbicide within the cell membrane disruptor mode of action reacts within the plant to form compounds such as superoxides and hydroxyl radicals, which then destroy cell membranes. The herbicides are typically not translocated, and the herbicide only affects areas of the plant that it contacts. The herbicide families within the mode of action include diphenylators, aryl triazonin, and so on and so forth. You can look at including the gramoxone, which is the paraquat. So you can try to look at this, look at the notes and try to understand this mode of action. The injury symptoms can occur within 1 to 2 hours after application. So these are appearing first as water-soaked foliage, which is followed by browning, which is a necrosis of the tissue wherever the spray contacts the foliage. Symptoms will appear most quickly with bright sunny conditions at application. Drift injury will appear as speckling on leaf tissue. Injury from soil applications or residues appears as a motile chlorosis or necrosis. Stop the video, pause the video, and look at this, all the information. Okay, to be continued.